Thank you very much, Wilhelm, for agreeing to give a Build a Cell seminar, and you can get started.
Well, thank you, Kate. Thank you for having me on the weekly Build a Cell meeting. My name is Wilhelm Huck. I'm a professor of chemistry at Radboud University in the Netherlands. And I'm part of the BASIC consortium, which is trying to build a synthetic cell. So I'm a chemist by training, but in the past year, 2 years, I've become sort of very interested in AI and machine learning as a tool to accelerate research in the chemical sciences. And I think it's also a very good tool for trying to help us building a synthetic cell. And so that's the topic of today. So there's a bit of results, but mostly it's just ideas and proposals and wishful thinking. So bear with me. Also a little bit thanks to start to Kees Dekker and his team who provided this beautiful image. So we start off with a beautiful slide. I guess it will go downhill from here because my artistic skills do not match Kees's skills at all. Right, there we go. Building a synthetic cell with a bit of AI assistance. And then just to remind you, what are we trying to do? Let me check the slide goes to the next one. Yes. So the BASIC consortium in the Netherlands is a large consortium. It's been running since 2017. It will run for 10 years, and we're already thinking about follow-up funding, as one does, in these large consortia. But it is really sort of trying to build a synthetic cell from the bottom up. And the fundamental questions that we're trying to address is really, can we go from lifeless to alive? Very much the previous slide of this sort of arrow going upwards. And we're very much interested, or at least I'm very much interested in the exact nature of that transition. How does this look like when you go from lifeless to alive? Can you be a little bit alive, or do you go from fully lifeless to fully alive? And is it like a phase transition, or is it some stepwise process? That's what I'm very interested to see if we can discover that very point. In general, you could say, well, I could also try and do the same thing from the top down, but I think if we try and do this from the bottom up, then I hope at least it gives us control and mechanistic understanding by building from known components and going ever more complex. But the real challenge that we are now starting or having to start to address is how to integrate millions of components. And maybe it's not millions of different components, but definitely millions of individual components that have to go together into that synthetic cell. And that is, I think, a challenge that we, well, maybe as a consortium, as a field, we have been underestimating a little bit, I think. And that's why I think today I want to pitch AI or machine learning in a very general sense to you as maybe a tool that we can use in the future. And so the key challenges, at least as I see them, in the field of trying to build a synthetic cell is that, of course, we have to build certain modules with functionalities like cell division, maybe ATP production at a certain rate, controlling cell-free gene expression, lipid synthesis, etc., etc. So that is challenging, but I think we are making progress. However, there are many functionalities for which we do not really have a blueprint. And then I really think about, say, homeostasis or robustness, or in a very practical sense, if you want to make an oscillating reaction network, we know how to do that, but I cannot control the frequency of that network. And there's also no design, no blueprint that tells me that I should add this, that, or the other feedback loop in this kind of ratio. So there's nothing out there that allows me to build complex functionalities that really are beyond the design capabilities of what we have at the moment. And then the integration of modules where, say, you have optimized the gene expression for a number of different genes, and then you want to optimize ATP production and maybe lipid synthesis. That is all good and well if you have to do that in an isolated module. But if they all have to talk together, then the integration is a very, very difficult at the moment trial and error process. And that is because there are many, many interactions in your system that you simply don't see, don't know about, but they put a certain load on the system. So think about it this way: these molecules, they're all in a very sort of small container in each other's proximity, and that means that they're all going to bump into one another, even if they have nothing to do with one another in terms of a chemical reaction. But when they bump into each other, they do make it impossible for that same molecule to bump into another molecule that is its natural reaction partner. And so therefore, our general concept of kinetics and rate equations that we learn in our first year in undergraduate school no longer holds, right? Because everything we know about general standard thermodynamics of chemical reactions means that we're in a dilute environment that is homogeneously mixed and remains constantly mixed. So that means that this integration of modules is actually a very, very challenging process. And so I think a solution to all of this could be, rather than relying on 100% design, relying a little bit more on some kind of the power of evolution. And of course, biology has relied on the power of evolution to get you to whatever you are at the moment. But in chemistry, we don't really do this, or in sort of experimental science, we don't really do this. And what I hope is that we can start doing this by essentially building a design, make, test, analyze cycles. And so for functions where we don't have a design, we could actually define exactly what kind of function we have in mind. And if we then say, well, we can use sort of high throughput experiments, we can use algorithms to access a very large parameter space, then maybe we can build this active learning loop that is almost like a laboratory version of evolutionary processes. And so let me so in engineering, this is called design, make, test, analyze cycle. I call it AI-assisted evolution, if you wish. And so it could look like something like this. So you start with saying, well, if you want to sort of make the correlation with evolution a little bit more complete, you say, well, this is my genotype, and my genotype is not just genes. My genotype is everything that goes into the design of my reaction. So all my input parameters, and they can of course be genes, and they can be proteins, and they can be lipids or different modules, etc., but it also includes concentrations of everything, volumes of everything, order of pipetting things together, maybe the day of the week, what you had for lunch, whatever you think is important has to go into this sort of input parameters, and that is your genotype. And what you then do is you simply do a lot of different experiments. So say 96 well plate, 384 well plate, the more the better. And then you look at your outcome of your experiments, and you're not looking for a perfect experiment that you say, well, I designed everything perfectly and now I want things to work. The only thing you say is, I have a certain objective in mind. So anything that gets me a little bit closer to my final objective, and let my final objective be something like a liposome dividing. So anything where you see a liposome even deforming a little bit means you're on the right trajectory. So what I need to do is I need to have a high throughput way of at least measuring something essential in this sort of 384 well plate. And this can be some fluorescence measurement, turbidity, patterns and shape, temporal patterns, anything. And what you then do is you feed all that information back to an algorithm that says, well, if you think that this and this well in your microtiter plate got you slightly closer to your objective function, then we give that a little bit higher sort of marking than all the other experiments. And that information goes back into the algorithm, which will then predict to you which other parameters you should be trying that will then maybe, hopefully, if you repeat this cycle, get you ever closer and closer to your final objective. And I think if we can close this loop and we can do this at sufficiently high throughput and for a sufficiently long time, then we are mimicking essentially what evolution does in biology, but we're mimicking it in an experimental wet chemistry laboratory setting. So that's what we're trying to do. And I will show you a couple of examples today where we have been starting to use some of these tools, and with a little bit of success so far. But at least I hope I can convince you at the end that this is maybe the way forward. And so I will show you one example of using machine learning to optimize cell-free gene expression. And I will show you another one using active learning and optimal experimental design to obtain complex reaction networks with correct function. So let me go to the first one. This is not our invention. A couple of years ago, Borkowski et al. Published a paper in Nature Communications on the optimization of cell-free gene expression. And they use a active learning algorithm to sort of improve the yield of cell-free gene expression. And you would say, well, that's fine. We know how to do this in the lab because we play around with magnesium concentration or maybe the sugar concentration, or we switch from glucose to maltose, or we change a parameter and we try and optimize it. That is not what they do in this paper. They have an algorithm. I'll talk a little bit more about this in a minute, where they can change many parameters all at the same time over a wide range of different values. And if you just say, well, I just have 11 different parameters and 4 different concentrations, then in principle, I have already more than 4 million different compositions that I can try. And for every one of you who has actually worked with cell-free gene expression, this is only part of your feeding buffer, right? So there are many more components that go in there, and you can actually vary them over a larger range of values. So I can make my parameter space actually quite a lot larger even than this. But what they show you here is that the algorithm slowly, after a number of different cycles, generations if you wish, starts to pick things up, and then it levels off. And they see a modest increase in cell-free gene expression. This was then picked up by Toby Erb's group in collaboration with the same group and published a few years later, again in Nature Communications, where now they have made actually a really nice interface. It's called Metis, and that's what we have been using as well, which is already in a Google Colab notebook, where the first thing you do is you define an objective function. And for most of us, that means in general, you're first of all interested in yield, right? You want to make as much protein as possible. But that doesn't have to be the case. You could also say, I want to make as much protein as possible given a certain rate of protein expression, or I want to minimize the amount of DNA I use and still get the most protein out, or I want to have an even production of protein regardless of what other parameter I'm changing, because I'm looking for a robust gene expression system, not for a high yielding one. So there can be many different objectives that you define. What you then do is you say, well, okay, what can I actually change in my experiment? So what kind of parameters do I want the algorithm to pick up on, and over what range of values do I want to vary those parameters? And that goes into your algorithm that will then give you a certain output that you can visualize, analyze, and then you go around this loop. Now, where is the AI part or the machine learning part? That is actually in the software underneath that is actually using a so-called XGBoost algorithm. And I'm not going to discuss that in great detail, but an XGBoost algorithm, you don't have to invent this yourself or train it yourself. It's actually, you can download it or access it via your Python library. And what XGBoost essentially does is it says, well, it only uses weak learners. So every machine learning model that is in there is actually not very good. It's almost a little bit like a random guess. It guesses whether your potassium should be higher or lower than 100 millimolar. Well, that is not very good. But if you essentially can have many of these weak learners all concatenated together, then every little guess that goes in the right direction makes a better and better and better guess, essentially. And so then it becomes actually quite powerful. And so if you're building a tree, essentially, and this tree of regression, the regression tree maps the input data point that you have on every leaf of the tree to a continuous score. So it can be potassium concentration, plasmid concentration. So everything that you put in the previous slide in terms of the parameters that you want are sort of leaves on the tree. And you're going to see for each of those leaves, is it the right value or the wrong value, the right value or the wrong value. And you're trying to then, for all the leaves together, you're trying to find the best possible outcome, essentially. That gets you closer and closer to your objective. So there's underneath, there is a regression model. I'm not going to talk about this in detail. There's a lasso regression and another regression, but that's not so important. What it essentially is going to do is it's going to give a penalty for everything that is a little bit wrong, or that makes things too complex, and it's going to give you a boost for everything that you sort of have correctly guessed in a way. Yeah. And so that way, you do this iteratively, and you essentially get to have a better prediction. So that's what we have used in, well, Metis uses this, we are using this as well. Just giving you one recent example where we have done cell-free gene expression, but then in an organism that has been exceedingly difficult. We were interested in working with the Craig Venter Institute in La Jolla to say, well, if we want to make a minimal cell, then the real minimal cell that we know is JCVI Syn3A. So if we want to build a synthetic cell in the most simple form, we should be able to use this organism. But everything we tried for many years, we didn't manage to make anything functional. And that is because the mycoplasma has some very nasty and very active nucleases that chew up your ribosomes and everything else that is in there before you can even isolate your lysate. Until eventually Andrei Sakai, who was the first author on this paper, found a way of lysing cells in a very effective way, which is called the nitrogen bomb method. Essentially, it means that you put your cells in a little stainless steel vessel, you put it on a very high nitrogen pressure, and then very rapidly you release the pressure. So essentially you're generating lots of bubbles also inside the cell, and the cells essentially explode when the nitrogen bubbles form. And that forms a sort of, that separates the membrane fraction very rapidly from the lysate, and we think that that means it leads to a situation where the lysate is no longer or very low nuclease activity. And then what we use is this Metis with the XGBoost algorithm, and we said, well, we want to now see if we can express, in this case, neon green. And you see in the beginning, it is all very low expression, although occasionally you have a little bit higher expression, until the algorithm starts to pick up on certain parameters. And then you really rapidly start to increase the yield of your fluorescent protein, getting us to the stage where at least we could see robustly, repeatedly neon gene expression in this very difficult and actually not so very active lysate. So that was a very good step. We are now expanding this a little bit, where Leonardo Morini, who was the first author of this paper that will come out soon, said, well, can we also sort of expand this method to gene expression from chromosomal DNA? And you might say, well, there's no difference, because if you have just plasmid linear fragments of DNA, what difference does it make? Well, the idea that we have is that if we want to go to a synthetic cell, we really need a very large piece of DNA, a synthetic cell chromosome. The problem with a chromosomal DNA is it's a huge molecule. It's completely wrapped up and entangled on itself. And so it might well be that the standard conditions that you normally use for gene expression from plasmid or linear DNA no longer work, because the conditions are such that in that case, your gene is simply not accessible. It wraps up on itself. And so we sort of do indeed encounter this. If you just add E. coli isolated chromosomal DNA to your standard mix of gene expression lysate and the different conditions, then what you will find is that it is actually not very active. By doing many rounds of Metis, we eventually settled on something that was much better performing. And then you say, okay, it's much better performing, good for you. You managed to, it's an order of magnitude better, roughly. But you could say, well, that seems like a black box, right? Okay, somehow it's gotten much better. But the advantage now is that I can, in principle, go back and retrace my steps. And each of these steps, I can go back and say, well, what was Metis actually doing? What did it pick up from all these different sort of experiments that I did? And what kind of trends did it pick up? And what you see is that it seems to be picking up that every time when I was doing my different experiments, it was suggesting it gets better and better if I have no potassium in my mixture. It also seemed to identify that my magnesium concentration should be around 8 millimolar, which is not so atypical. But it is really surprising that you should go to only 1 millimolar or no potassium. That was really surprising. And it sort of, you can then use this to say, well, what is really happening in your system? And so when we were investigated in much more detail, what we find, and again, read the paper when it comes out in a couple of weeks, hopefully, is that actually your balance between transcription and translation starts to shift. You start to see where the limiting factors are in your cell-free gene expression when you're doing it from this chromosomal DNA. And when you then look at the numbers and you look at the optimized system, then actually it's an interesting sort of calculation that you can make, because now, in the end, the other thing it found was that we have to lower the lysate concentration. Sorry, not the lysate concentration. We have to lower the genome concentration. So we're ending up with concentrations of the gene that are in the picomolar regime, whereas normally when you do cell-free gene expression, you're in the nanomolar regime. But yet we express the roughly similar levels of GFP, roughly low micromolar levels. But it does take a much longer time, 10 hours instead of 1 hour. So if you look at the numbers of proteins produced per gene, it is really much more efficient. If you look at the amount of GFP produced per gene per minute, it is also a lot faster, actually. So now we're beginning to see an insight into the dynamics of what appears at first sight a simple system. You go from DNA to RNA to protein. But in this complex mixture where you have all these different proteins in the lysate, you have all these different salts, there's actually a lot to study to really fine-tune what is going on in these kind of systems. And to cut a long story short, even if you now look at these best performing systems, there's still an order of magnitude or two of the actual biological system, which is still much more efficient than anything we can do in the lab. So there's still plenty of room for optimization. All right, that was example number one, where we were looking at active learning to, XGBoost and sort of algorithms to optimize and move through a very large parameter space. The other one is about optimal experimental design. And the thing we are doing there is to say, well, there's lots of sort of modules that we want to make, but we simply don't know how to make them, because whenever we put them together, we have sort of parts that activate and parts that repress, and we think we can build a functional system, then it doesn't actually work the way we would like it to work. So an oscillator doesn't oscillate, a switch doesn't switch, etc., etc. And again, I think we can use active learning and optimal experimental design to sort of solve that problem. This is a very long story. I can only highlight a few things. Look it up. It was published in Nature Communications a little bit over 1.5 years ago. And so what we're going to do here is that, as I said, the design of functional models in practice doesn't work very well. They never give you the right functional output. And we thought that if we can make a library of individual parts and then have a rapid method for see how they all interact with one another, and then have a model that can sort of build from build a network based on actual data that come from the sort of parts in action in a network, we can actually design, or we can actually sort of move towards functional networks much more quickly. So what we did in that case is we built a library of parts to build a incoherent feed-forward loop. So a very simple one. We have a couple of promoters, we have a couple of operators, we have a RiboJ section, we have a ribosomal binding site, we have 2 different reporter proteins, 3 different repressor proteins, and we use different sigma factors to play around with the strength of gene expression. And so you can make lots of different repressor constructs. So the overall thing is the incoherent feed-forward loop that we can study in itself. And so what we have an underlying mathematical model that is simply based on rate equations, where we know how strong repressors bind to promoter sites, how strong the sigma factors sort of lead to transcription. We know the rates of transcription, we know the rates of translation, we can measure the rates of maturation of GFP, we can assume degradation rates. We can try and build this model as complex and complete as we want. We do assume, though, that there's no competition for ribosomes and core RNAP, but in principle, we can expand this model as complex as we want. Now, what you will find in general is that this is not enough to really predict how the incoherent feed-forward loop works. So we will have to do a lot of experiments. And we're going to do this in a flow reactor because we have to actually do so many different experiments that we cannot do this in a 384 well plate. So what we do is we have a microfluidic reactor where we have 8 parallel reactors. So in here, we have everything reacting. We can visualize, we can image the reactor and measure the fluorescence so we know how much gene expression is present. And in each of these individual channels, we can change the concentration of input concentrations of different salts, but also different concentrations of DNA fragments. So here's where we have full control. And then overall, we have full control over the flow because we can open and close using sort of fluidic valves in which channel what kind of concentration will flow in and will flow out. Okay, so this is a complex reactor that takes a long time to design and build. But once you do this, you can actually sort of say, well, now I can use a flow pattern in my microfluidic reactor where I have 3 different feed-forward loops using 3 different repressors, and I can have different concentrations of reporter, activator, and repressor genes that are constantly changing. And these flow patterns are designed by an algorithm that says if you use these kind of concentrations as input, you get maximum amount of information from the output that you get, because the output is only this fluorescence that comes at the very end of my reaction. So I need to have this sort of information that I get. And as I said in the beginning, I'm not interested in characterizing the full experiment, right? I want to have key information, and I want to use that key information to deduce what is going on in my reaction mixture. So this is my output, this is my input. If I do this, then what you will find is that very rapidly, my error margins on all the different steps in my reaction mechanism starts to shrink. So my model becomes ever more deterministic of the actual system that is happening in my reactors. And when you then do this, you can say, well, now, because I can now build a much better model of the actual dynamic of the gene network, I can say, well, I can design a bistable switch. I can then predict what kind of profiles I should put in to sort of for my DNA fragments, and I can predict what kind of output I should be getting from my fluorescent reaction. So it should switch between red and green, so M-cherry or DE-GFP. And when you just try this with an unoptimized model, you will never find the regime where this thing actually switches from red to green. In our case, because we use this optimal model that actually understands how these things interact when they're all in this mixture, it actually predicts pretty accurately, even quantitatively, what your switch behavior should be. So that is something that we tried a couple of years back. We have now been expanding this, skip this slide, and we are expanding this to metabolic networks where we use a similar kind of algorithm. Now we have a metabolic network, and you say, well, what do I want to know? I want to know, actually, I'm producing UTP, GTP, ATP. But when I want to build a synthetic cell, I want to have control over the ratio of UTP, GTP, ATP that I'm going to produce. Otherwise, I might just be producing UTP, which I don't really need because I need much more GTP and ATP, for example. But if I want to control this, and these are all kinds of coupled reactions, it's very difficult to predict what I should be doing to get some kind of well-defined ratio of these 3 different outputs. So this is a network that is based on the nucleotide salvage pathway. We implemented this network by immobilizing the enzymes on beads. Then we have 6 different inputs, which are the small molecules that go in. And we have 8 different species that we measure coming out at the other end. What you then do is you use the algorithm again to now have a particular input profile for each of the different sort of inputs that I can give. And then we measure here the concentrations of our output. So the triangle values are HPLC-derived data points for all the different nucleotides that I get out. And the solid line is the predicted concentrations that I should be getting. And as you can see, this is after 2 iterations, I get a very good predictive model now. So my model is able to predict what the concentrations will be in response to all these different input concentrations. And you can use this to say, well, now I can start to predict what kind of input concentrations I should be using to actually predict what output concentrations I would get. So if I say I want to have ATP, UTP, GTP concentrations in a ratio of 3 to 1 to 1, or 6 to 1 to 1, or 1 to 2 to 3, or any other combination, I can actually predict what kind of input concentrations in the model I can use to accurately get these kind of output predictions. I say accurately, it doesn't do very well if you're in a very low concentration regime, and that is probably because our HPLC is not sensitive enough to pick things up in that low concentration, so we are way off in terms of the model. So this is sort of very quick bird's eye overview of how I think we can be using AI tools. Broadly speaking, I'm talking about regression methods, optimal experimental design, active learning, maybe also image recognition and those kind of things, and high throughput experiments to allow us to explore the vast parameter space that we need to traverse in these kind of experiments. And the second bit is that if we can do this, then we also can start to think about making functionalities for which we don't actually know how to do this. And robustness is a good example. How do you make a synthetic cell that will sort of still function if you change the temperature by 10 degrees, or if you get your concentrations of, say, potassium off by sort of 10 millimolar? It has to be robust. And so how do we design these things? We don't know, but by combining these kind of AI tools and high throughput sort of high throughput experiments, we hopefully can just discover them in a practically relevant fashion. So mind you, when I'm talking about AI, I'm not talking about sort of ChatGPT or DAL-E or these kind of tools, so the generative methods. I think it's a bit too early for that. We don't really have enough data to train these kind of very data-hungry foundation models. That is something that will come in the future, but that will take another 5 years or so, I think. So with that, I wanted to wrap up. I want to thank the group, and I want to especially thank BASIC for funding this work. Big thanks to Andrei, who did all the work on the mycoplasma, Leonardo, who has been doing all the work on the expression from chromosomal DNA. Bob has helped both Andrei and Leonardo implementing the METIS and XGBoost algorithm. And Bob and Roel also have pioneered the use of optimal experimental design, where Roel did the gene expression networks, and I should have put Tao Zhou here as well, who worked on the implementation of the metabolic networks. And with that, I come to the end of my talk, and I'm happy to answer your questions.
Thank you. Thank you so much. There are questions in chat. Can you see chat?
Okay, this gives me a little bit. Okay, we can go through the chat. Let me start on the top somewhere. Okay, I'm looking at the question about resource competition.
If you could please read a question, because people watching the recording will not see the chat.
Absolutely. So Tae-Sok Moon has the question that for practical applications, resource competition plays a major role, limiting the system's performance. To me, your work gives great insights into the biological phenomena, but for practical applications, it may need to be revised. Could you comment on this point? Actually, Tae-Sok, you're quite right. Research competition does play a major role. So in many examples, you should take this into account. And this is the beauty of the system. So what I've shown you is a loop where essentially you do optimal experimental design, where you say, this is my model, and I'm going to do an optimal set of experiments that will find the kinetic parameters for my model. Now, what Bob has been doing in the background to say is, well, the second thing is you now say, instead of just choosing parameters, you're also going to choose models. So in principle, I could give it 1 billion different models, and it is also going to select which model will sort of best fit the data, and what kind of experiments should I be doing to best check which model would be suitable. So this can indeed be done, but the computation becomes quite, well, demanding. So for the cell-free gene expression, it's challenging. For a little bit smaller network, it becomes more feasible to do this. But you're absolutely right. Resource competition is important. And yes, we can explicitly take this into account into the model. There's a question from Harrison Daniel Sambor. Could you apply the system to other than E. coli extracts, mammalian, for example, or is there something specific to E. coli in there? No, not at all. This is not specific to E. coli. What you do want to make sure is that you have a robust system. So if you get irreproducible results, everything breaks down. So what you see in many cases when you do cell-free gene expression and you do it with a different lysate, for example, it works, but you don't get exactly the same yield. And that is very bad, right? Because your algorithm needs a certain baseline. It needs to be it needs to be able to trust your previous data. So if you cannot reproduce your data, you have a serious problem. So that's a major problem. And the other thing is that you need a starting point. So for the mycoplasma, it was extremely challenging in the beginning, because if you have 0 gene expression, then there's nothing to optimize. So that is another problem. So 2 challenges, reproducibility and a certain lead. You have to be above 0, and then you can start optimizing. Is there anything, so Emma Bernard asks, is there anything we are still missing on the AI side? Could you use generative tools once you collect all this data, feedback loop of some sort to train the AI? I think there's not really something missing for what we're doing at the moment. So the algorithms, XGBoost and Metis are well established. The optimal experimental design is something that we have home built, but there's other people who have built other home built algorithms. For that, you don't really want to design something yourself, because it's very difficult to build new algorithms, or at least I'm not a mathematician, so for me it will be very difficult. And at the moment, generative tools are a little bit out of reach. And the reason for that is we don't nearly have enough data, right? So if you think about large language models, ChatGPT was trained on, I think, 150 billion different data points. GPT-4 was 450 billion data points. More advanced models go over 1 trillion data points. If you think anything we do in the experimental sciences, if we do 1,000 to 10,000, it's a lot already. So we're not even remotely in the same ballpark. So we cannot do that. So the way you would have to do this at some point is say, well, somebody trains the foundation model, and a foundation model is like your ChatGPT, your large language model. And what you do is you retrain, you fine-tune your ChatGPT on your language that you're interested in. So suppose you have ChatGPT, I feed it with all the metabolic networks of all the organisms that I can find, and I tell ChatGPT to learn the language of metabolic networks. So that is something that you could try. We haven't done it, so I don't know how difficult this will be in practice. But this is something that you could try and then hope that ChatGPT starts to understand, uncover what is the relationship between different nodes in these networks. And then if you ask ChatGPT to suggest a new network with certain different properties, maybe it will come up with something. But this is far away. And for us, this is not something that we want to attempt right now, because I think this is also maybe not necessary at the moment. So if we can build this active loop where you do lots of experiments, you pick the best ones, you feed them back into the algorithm. If we can automate that, then I think we're already sort of orders of magnitude advancing from what we're doing right now. Take One had a second question, so I'm going to skip that first and give Dieter. Dieter Brown had a question. Playing devil's advocate, Toby Erb, when pushed after one of his talk, was clearly saying that the selection they used AI on was easily done and not worse by human insights. Even if it's more complex, multi-parameter optimization has been around for a long time. Just check numerical recipes and see, correct? So honestly, what is really different from that to AI, and not to forget that in multi-parameter optimization, you're getting error bars for your experimental parameters upon optimization. I think, well, first of all, if you do multi-parameter optimization and numerical methods, you do need to have a very good model. In my case, I don't necessarily need to have a very good model, I think. So maybe, well, Dieter is saying no, so I'll give you the chance in a minute to respond to that. It is not necessarily better or different. I think it's cheaper and faster to do it this way. So I think it's tied into experimental data. And if you can go around the cycle quickly enough and often enough, you converge on your objective point faster than trying to parameterize your model. I think the parameterization will be the most challenging bit, especially also there. You don't know if you're using the correct model, right? So that's where I think is that this is a sort of faster way of getting there.
Can I chip in shortly?
Yeah, okay? Absolutely.
I mean, these optimization models, they have no model behind, but they go iteratively in each step and give you the next best check for your next experiment in a very even nonlinear landscape. So they cope with that. They could. I thought almost they could give you a better estimate for the next experiment than.
But that is what we are essentially doing in the optimal experimental design part. So the second part was showing you Bob van Slijs's work, where we do the nucleotide synthesis or the gluing together of the different elements of the feed-forward loops. That is essentially what you are suggesting. You essentially suggest a whole series of experiments that you can do to give you maximum information to fit your model. And if your model fit, then you can sort of start to predict how the system functions. So there you do rely on a lot of calculations and input into your model. With the Metis and the active learning there, the XGBoost, there you don't really have an underlying model. There you simply need to rely on sufficient number of experiments and the right parameterization, so choosing the right parameters.
I mean, we can discuss longer, but my question was more, if you really ask an informatic numeric optimization guy, they might come up with an algorithm far away from AI, which is better because we're not dealing with millions of data points here.
I think for your cell-free gene expression, you don't have a model. So you can predict sort of so you can numerically analyze your model and come up with experiments. It's just that model doesn't describe what is going on in your system. And so you will not be able to predict under a wide range of conditions. Dieter and I am going to have a beer on this.
That's fine. That's fine. At some point, I just, these optimization, they don't need a model basically. So, but we might talk.
Okay. Taekmoon had a second question. Taekmoon. In addition, what would be the remaining challenges for your great tools to enable real-world applications? And depends a little bit on what you mean with real-world applications, in the sense that we are optimizing the expression of an actual lysate. So that means, to me, that is the real world. If you think about commercial applications, I think that is different. It depends on what people are trying to do there. So maybe you can.
I'm talking about potentially, commercial application later on, because I see huge potential from your approach, and I'm an engineer, so I want to apply your tool to make something happen in the real world, and that's actually my question.
Yeah, that's a good question. I don't know. So especially for the enzymatic networks, for example, we did in fact talk to various industrial people. The problem is that I think industry doesn't use such complex reaction networks in an industrial scale. So often they use one or two enzymes, and they simply maximize their turnover. So it's just running at a Vmax. And so then you don't need such a difficult algorithm to optimize a complex network. So to be honest, I don't know where you would use it in industry at the moment. Yeah, so that's the short answer.
Yeah, makes sense. Thank you.
Gersje Koenderink asks, why can you get away with much fewer experiments per round for the cell-free expression than for the enzymatic network systems? Simpler genotype. Well, it's a different method, right? So for the enzymatic networks, you have a full model in principle, or you have many models, as I was discussing earlier with Huck. So you can have many competing models, and you're trying to find the kinetic parameters and the model that describes the system best. So then you do need a lot of parameters, a lot of experiments that decouple all the interactions in that networks that are correlated. So if you think about an enzymatic reaction, then if you say, I have a higher turnover on my reaction, it could be that you had a stronger binding of the substrate to the enzyme, or you had a faster K-cut. So it could be Km and K-cut that you could change, but they could basically account for each other's change. So to discover whether it should be K-cut or Km, I have to do a number of additional experiments to make sure I'm in a different regime of the enzymatic activity, essentially. So with the methods and the optimization of the cell-free expression system, I don't really have an underlying model. I'm simply trying to find a sort of path through my parameter space that will match my objective function best. And so at every step, I'm simply trying to minimize the difference between the experiment outcome that I have and the desired outcome that I would like to have. And so there is a different way of, that is simply a different way of doing it. And it's a very efficient way of going through a huge parameter space. Now, the algorithm doesn't give you the model of what is happening inside, but you can, as I've shown, you can trace back what are the key parameters in the system that give you, that have the most impact of reaching that objective function. So there's a different approach to what you want to know about your system. James P had a question. Hi, James. Beautiful talk. Might you know the state of using physical models to predict rates or relative rates, for example, to use molecular dynamics simulations of the interaction between transcription factors, RNA polymerase, and DNA? Yeah, in principle, I would always naively say, well, you should be able to do that, right? I can build the model and I can predict, and in practice, it never works. So that's our main challenge. And this is partially due to resource competition. It's partially due to these models not sufficiently capturing what is actually going on in your system. So you try and make the model as simple as possible, but when you say already when you're doing sort of RNA polymerase, are you just going to consider that as one binding step, or are you going to take the holoenzymes into account? Are you going to take conformational changes into account, allosterics into account, or do you just say, no, it's a mass action kinetics, it just binds, bang, goes on? And so you already see that the more complex and more accurate you make your model, for example, using molecular dynamics simulations, it blows up, right? Because then there's so many parameters. That means you have to do so many experiments to know what these parameters are. It becomes impossible. So it's this balance between stripping down the model to something that is simple enough and predictive enough to tell me what is going on in my experiments, but not simpler than that. So that's the way we approach it.
Cool.
Let's see, other questions?
I think you are. I'm really curious about the optimization questions, but I guess you answered that pretty exhaustively. But the one follow-up question I have, though, is you said it's impossible to have those millions of data points. What about microfluidics? Is it possible to set something up like a tiny droplet system? Some of those systems that continuously change concentration of one reagent, then another, then another.
Yes, that's very good. You should be doing that. The problem is then you still have now shifted the bottleneck a little bit, right? Because now you say, okay, I can do many experiments by making many droplets that have each a slightly different composition.
Yeah.
Now, how do you know what's going on in these droplets? So I still need to be able to measure something, right? So, and the problem then becomes the more complex your system is, and the fewer observables that you have, the more difficult it becomes to essentially correlate sort of input and output, so cause and effect. And so the advantage of using the microfluidics as we have done twofold. For example, you see in the experiments where we did the gene expression networks, then you only have the colored proteins that gives you the readout. So you don't really know very much about your system, right? You don't measure the RNA concentration, for example. You only measure the endpoint. In the case of the enzymatic networks, I measure 8 different species. So I have already 8 observables in my system. So for each experiment, I get a lot more information about what is happening in the system. And that is also something that you can do with the methods and the sort of algorithms that allow you to do gene expression optimization. If you only measure protein yield, then again, your algorithm can only sort of build the next round of experiments based on protein yield, because that's what you measured. If you give it more information somehow by measuring something else at the same time, it could be anything, right? Change in pH, potassium concentration change, anything else that is sort of informative on the system, your algorithm will do better. And so I think when you sort of try and set up this loop of evolution in the lab, essentially, the more you can measure on your system, the better. But it has to be at a sufficiently high throughput, because otherwise it will take you forever to fully characterize everything.
So,
Yeah.
Kate, a question from Oxford here. What about the Rumsfeld problem, the unknown unknowns, particularly when it comes to creating life? I mean, the Ventus method of chucking things out, you're not going to miss anything. And say for cell-free systems, for example, maybe rubidium was the answer, and it was never in there, right?
Yeah, I can see your concern in the sense that if you treat it as a kind of a black box, then you run the risk that you have a perfectly functioning system, but you don't really know why, and it becomes very difficult to understand why. Actually, with these stepwise sort of, well, with a laboratory-assisted or AI-assisted laboratory evolution, you have the advantage that you do this in separate generations. And for each generation, of course, you get the full data set, and you get a logbook of what methods or what your XGBoost algorithm is trying to correlate. So you can go back in and retrace the different steps that got you to this optimal composition. And then, of course, that still doesn't tell you immediately why things are happening, but it gives you a smoking gun. It tells you that apparently if I change those parameters, it got me to a particular point. And then you need to use your biochemical insight and Leonardo, well, you have to become a detective essentially and say, well, if that is my smoking gun, then really what is happening in the system that causes my gene expression to be so much more efficient than it was when we were just doing plasmid DNA. So yes, it is not an immediate output of the algorithm, but I think the method, the workflow does actually allow you to go back in and reinvestigate.
Yeah, no, I guess my concern is that particularly at the higher end, when you're trying to generate a living system, is simply that you're putting known entities into your experiments. And what if, I'm not talking about sort of vitalism here, but let's say there's just simply something that you're just missing out on.
Okay, you really want to go to the unknown unknowns.
Well, if you do the top-down approach, right, all the unknown unknowns are in there floating around.
I think you're quite right. I don't think we will pick them up. So the AI might figure that there is something happening, and it will somehow control those parameters by pushing on others that I can change in my reaction mixture. But I think with the current methods, it would not allow me to see those.
But it might tell you there's something missing, right? To give you more credit, right? If you're saying you're 10 or 100-fold down on a natural system, it's giving you a clue that there's something missing.
Yes, but the clue as to what it could have been, then I have to start searching among the parameters that I did change. If it's a really unknown, then I'm just thinking, nah, how would I be able to sort of get on the trail of something like that? Because it depends a little bit if I can see it essentially in some of the other parts of the system. I would need to think about how to do that. It's definitely not necessarily initially in your design at the moment, because you're kind of agnostic to what the underlying model is. you can trace back to see what the system has been doing, and then you have to use your existing knowledge. So I would need to think about an actual example on how that works.
No question I can think of. So.
And I think it's going to get really interesting once the evolution take over too, because then you're going to have unknown unknowns that we cannot predict ahead of time because they don't exist yet. Not that they're part of the system right now, they will arise later.
I think, Kate, if your synthetic cell shows uncontrolled evolution by itself, then I think it's time to probably pull the plug on it, because then you really don't know what you're doing anymore.
I already don't know what I'm doing, so I'm not that far from it. All right. Any more questions? Okay, thank you very much. Thanks, everyone. Thank you so much, Wilhelm, for a great talk, and have a great rest of your week, everyone.
Thank you. See you all. Bye.
Bye.